there was a benign cyst on my removed, but because of the COVID-19, it's really hard to get the surgery scheduled. So thanks for bearing with me. I'm very pleased that I'm being considered as a candidate for the faculty position at the University of South Carolina, and it's my great pleasure to be able to have this opportunity to share my research on EEG signal processing and biomarker discovery in an effort to achieve precision mental health. I have been working with EEG for more than 15 years, and I'm always fascinated by how much information we can get out of EEG and how much we can push the limit of this classic brain recording technology in translational research. So here is the outline of my today's talk. I'll begin with challenges in precision mental health and EEG signal processing, and then I'll walk you through my research work conducted to address these challenges in 3 parts. And finally, I'll briefly discuss my future research plan on EEG-based biomarker development platform in an effort to refine diagnostics and optimize treatment. Psychiatric disorders are leading contributors to the global disease burden. Depression accounts for $100 billion in healthcare costs, and PTSD accounts for 60% of VA's health spend. Despite such an enormous spend on mental health, there is actually very little progress that has been made in treating psychiatric disorders. The fundamental issue is that psychiatric disorders are diagnosed based on combinations of symptoms, and patients with the same diagnosis may actually fall into biologically heterogeneous subtypes, which may require different treatments. However, today, there is no approved biomarkers that allow us to objectively track, diagnose, and treat psychiatric disorders and reliably dissect such biological heterogeneity. The entire process is fraught with subjectivity and trial and error. Over the past years, I have been developing technologies to drive research in circuit-based precision mental health, where the goal is to decode disease biomarker information from brain activity in a precise manner for treating psychiatric disorders. And to this end, I have been using non-invasive EEG to measure brain activity from sensors on the scalp and developing tailored signal processing and machine learning algorithms for decoding disease-relevant information from EEG. For those of you who are less familiar with EEG, EEG is the summation of postsynaptic potentials across many neurons, which occur when the neurotransmitters in the presynaptic neurons bind to the receptors in the postsynaptic neurons, essentially changing the flow of the ions across the cell membrane. And the reasons why we use EEG as opposed to other neuroimaging modalities to measure brain activity are severalfold. EEG is cheap, its setup is easy and fast. EEG is non-invasive and contains rich temporal information with millisecond temporal resolution. EEG also directly measures electrical brain activity, and as just described, is closely related to neurotransmission, which is important because the pathophysiology of many psychiatric disorders can be linked to neurotransmission, and many psychiatric drugs are targeting the neurotransmission process as well. So as a result of these advantages, EEG can be relatively easily translated to the clinic as a point of care biological test. However, decoding disease biomarker information from EEG is non-trivial, and the challenges are mainly on the analytical side. EEG is highly noisy. The background noise is typically more than 10 times stronger than brain activity. The spatial resolution of EEG is low. Reconstruction of brain activity from scalp EEG requires solving a highly ill-posed inverse problem. And there is also substantial variability between the EEG data of different subjects. Models trained on a portion of subjects may generalize poorly to the others. So my previous research was carried out to address these data analytical challenges in the context of precision mental health and EEG decoding in general. Specifically, I developed the Bayesian models and algorithms for extracting spatial temporal patterns from EEG and for accurately localizing source activities. I also pioneered a series of spatial temporal filtering algorithms for robustly decoding EEG signals and also for selecting channels to facilitate the translation of these algorithms. And finally, in collaboration with many psychiatrists in the world, I was able to apply the models and algorithms that I developed to identify EEG biomarkers for psychiatric disorders. So next, I'm going to walk you through all these contributions in greater detail. First, the statistical modeling for EEG analyses. In many basic or translational studies, the goal is to identify brain patterns that can differentiate multiple conditions. In this typical example, subjects are imagining the left or right hand movement, and we would like to find the patterns unique to each one of these two conditions. It is well known from prior neurophysiology studies that when subjects imagining their hand movement, there would be a decrease of alpha band power in the contralateral motor region. For example, when the subject is imagining a left hand movement, there would be a decrease of the alpha band power in the right motor cortex. And similarly, for imagining right hand movement, there would be a decrease of the alpha band power in the left motor cortex. And this modulation of the EEG power is exactly the basis for motor imagery brain-based brain-machine interface. However, the example I put here is an ideal case, and in general, differences between conditions may not be this obvious in the sensor space. And in this case, we may need to transform the sensor space EEG to a latent space where the differences between conditions can be maximized. And the challenge is designing methods to learn spatial temporal decomposition for identifying differential patterns between conditions. For EEG oscillations, there is a popular discriminative dimensionality reduction technique called common spatial patterns, or CSP, that has been widely used for extracting differential brain patterns between conditions. And the differential brain patterns can be obtained by solving a generalized eigenvalue decomposition problem. However, CSP by itself does not provide insight into how EEG is generated by the underlying brain patterns. And CSP is also prone to overfitting because the parameter space is unconstrained. So in light of the pros and cons of CSP, we developed the generative model, which is essentially a coupled latent linear model where the mixing matrix is shared across conditions. Interestingly, the maximum likelihood estimates of the parameters in this model yield the solution to CSP. So this model not only provides a generative interpretation into CSP, but also opens the possibility of improving CSP by extending this model. So on the basis of this generative model, we use the Bayesian inference to automatically determine the number of latent signals. And this algorithm substantially outperforms CSP in terms of latent signal estimation. Event-related potentials, or the ERPs, are the brain response to the external stimuli. Although the waveform of the ERP is relatively stable, its amplitude and latency can vary substantially across conditions. And these two parameters provide important information about how the underlying neural processes shift across conditions. In order to accurately estimate the ERP amplitude and latency, we developed a mixed effect ERP model where the waveform of the ERP is modeled as a fixed effect, and the amplitude and latency of the ERP are modeled as random effects. We also developed a variational Bayesian algorithm called BEEP to estimate the unknown parameters in this model. BEEP is able to yield significantly higher accuracy for the ERP amplitude and latency than existing ERP estimation algorithms. Now, for source of localization of brain activity, a long-standing challenge is to estimate the spatial extent of the brain activity. To address this challenge, we developed a Bayesian source imaging algorithm called BESTIES to automatically determine the spatial temporal patterns of the brain activity. Specifically, a Markov random field is employed to model the spatial pattern of the brain activity, and a set of temporal basis functions are used to characterize the temporal dynamics of the brain activity. Also, to enable efficient model inference over the high resolution cortices, we developed a scalable version of BESTIES based on variation of Bayesian inference and convex analyses. We also show that many classic source imaging algorithms can be viewed as special cases of BESTIES, and BESTIES substantially outperforms these algorithms in localizing spatially extended sources in terms of smaller localization error and less spatial diffusion. Now let's move on to my second contribution, which is developing machine learning algorithms for EEG decoding. And these algorithms are suited for applications where we would like to map the EEG signals to a target variable. For single trial EEG classification, the goal is to assign to each single trial of EEG a specific class label. For different subjects, the discriminative information may reside in different spatial locations or frequency bands. Conventionally, the selection of EEG channels and the settings of the temporal filters are determined manually based on prior knowledge, which heavily limits the classification performance. And the challenge is therefore designing methods to learn individualize the spatial temporal filters in order to extract the discriminative information from EEG. We first developed an algorithm to optimize the spatial and temporal filters in an ordinate fashion. This algorithm is able to handle two issues. The first is the overfitting issue because of the high dimensionality of the temporal filters. And the second issue is that the cost function is highly nonlinear with respect to the unknown parameters when the temporal filters are considered for optimization. To address these two issues, we reparameterize the temporal filters in the spectral domain by leveraging the fact that the temporal filters can actually be diagonalized by the Fourier transform in the spectral domain, essentially converting the originally nonlinear cost function to a linear cost function. And based on this reparameterization, we employed maximum margin learning from statistical learning theory to regularize the spectral filters. And this spatial temporal filtering optimization can converge to a solution within a small number of iterations. Now, there is no guarantee that ordinate optimization of the spatial temporal filters can converge to the global minimum solution. To address this limitation, by making use of the structure of the cost function, we recast the spatial temporal filter optimization as a power ratio maximization problem, where a closed form solution can be obtained. With this reformulation, the solution can also be penalized via the L2 norm to prevent overfitting. So this algorithm yields significantly higher classification accuracy than existing EEG decoding algorithms on a number of EEG decoding datasets. This reformulation also allows us to design an algorithm to select channels for translational purpose. To do so, we converted the cost function to a new one, which is invariant to the scale of the spatial temporal filters. So there is no ambiguity of multiple solutions to the optimization problem. And then we impose a group sparsity constraint on the combined spatial temporal filters as a regularization term in the optimization problem to select channels. So this channel selection algorithm substantially outperforms existing channel selection algorithms in terms of the classification accuracy under a range of designated numbers of channels. And the selected channels also appear in reasonable locations aligning with neurophysiology. And finally, by building upon and extending the algorithms that I developed, I was able to discover a number of EEG biomarkers for psychiatric disorders in order to bridge the gap between precision medicine and mental health. At present, there are two predominant approaches for using neuroimaging data to dissect the heterogeneity of psychiatric disorders via machine learning. The first approach attempts to derive subtypes using unsupervised learning based on the neuroimaging data alone without referencing to any clinical information. And therefore, the derived subtypes are more likely independent of a particular treatment. By contrast, the second approach maps the neuroimaging data to the treatment outcome as a result of a particular treatment using supervised learning. And therefore, this approach is able to yield treatment-specific biomarkers and can predict whether a patient would be responsive to a particular treatment or not. So next, I'm going to tell you three biomarkers we discovered using these two approaches. And each discovery entails methodological innovations in EEG signal processing or machine learning. In the first biomarker study, we discovered a neurobehavioral phenotype for PTSD. Our approach to stratify PTSD draws on the premise that disruptions in basic information processing underlying cognition actually forms the foundation upon which various aspects of PTSD are built. Subjects enrolled in this study went through a battery of neurocognitive tests and then neuroimaging sessions, including both fMRI and concurrent TMS-EEG at the baseline visit. And then they received psychotherapy as the treatment, which is considered the gold standard treatment for PTSD so far. With the multimodal data from each subject, we then ask whether leveraging individual differences in information processing impairments could lead to new phenotypes within the broader clinical syndrome of PTSD. The PTSD phenotype we discovered is characterized by impairments in the delayed recall of verbal memory and a decrease of the resting state fMRI connectivity in the ventral attention network, or VAN. And this particular phenotype was identified in two independent and clinically demographically distinct populations of PTSD versus healthy controls, demonstrating this finding is replicable. We also assessed the clinical relevance of the PTSD phenotype by examining its relationship to both the baseline symptoms and its capacity to benefit from the psychotherapy treatment. We found that this combined phenotype with impaired memory and resting state fMRI connectivity is not associated with the baseline symptoms, but nonetheless could be used to predict a poor response to psychotherapy, demonstrating the clinical significance of this phenotype. To provide insight into the causal circuits associated with the discovered phenotype, we used the concurrent TMS-EEG to stimulate two brain nodes involved in cognitive and emotional functions, namely the VAN and the frontal parietal control network, or FPCN. The identified causal circuit response can then serve as a potential target for using brain stimulation therapies to remediate the connectivity deficit. However, an outstanding challenge is that the TMS-EEG data is highly noisy due to the enormous stimulation-related artifacts, and manually cleaning up the TMS-EEG data is both labor-intensive and subjective, which is especially problematic for our study involving many subjects. I still remember back in the days when I started my postdoc at Stanford, I literally spent a few hours per day going through the raw TMS-EEG data of a few subjects and then manually labeling the artifacts in the data. I got to tell you, it's definitely not a lot of fun to do it every day. So to address this challenge, we developed a fully automated artifact rejection algorithm called ARTIST for TMS-EEG. This algorithm is 20 times faster than manual artifact rejection and demonstrates excellent generalizability across populations, study sites, and EEG systems. Compared to the manual artifact rejection by experts with extensive annotation, EEG annotation experience, the clean TMS EEG data are highly correlated between the automated and manual artifact rejection in terms of different stages of the TMS EEG potentials. And by using the TMS EEG data cleaned by our fully automated artifact rejection algorithm, we were able to identify a causal circuit response associated with the discovered phenotype. Specifically, we found a pora within VAN fMRI connectivity is reflected in a more profound circuit perturbation when the right-sided prefrontal VAN node is stimulated. And the circuit perturbation actually takes the form of profound desynchronization in the alpha band EEG. So this finding is actually consistent with the rTMS treatment in PTSD, where the right-sided prefrontal region in the vicinity of our VAN node is typically stimulated, and clinical efficacy was reported. And further rTMS treatment optimization can be guided by monitoring the change of the TMS EEG response to the right-sided prefrontal VAN stimulation. And our study, therefore, lays a path towards the use of TMS EEG to bridge the gap between the biomarkers and therapeutics. The PTSD phenotype I just presented is a multimodal biomarker combining both neuroimaging and behavior. In the second biomarker study, we discovered transdiagnostic subtypes for psychiatric disorders based on the EEG data alone. And the feature we used to derive the subtypes is the power envelope connectivity, which measures the correlation between the slow power fluctuations of EEG oscillations. And in our earlier study, we have shown that EEG-based power envelope connectivity is able to yield brain networks similar to those derived from resting state fMRI connectivity, demonstrating that power envelope connectivity is actually a neurobiologically meaningful connectivity metric for EEG. And based on the power envelope connectivity, we developed an EEG-based subtyping framework for psychiatric disorders. Specifically, the resting state EEG signals are first bandpass filtered into different canonical frequency bands. And then within each frequency band, the power envelope connectivity is calculated between vertices in the source space. And then the vertex level power envelope connectivity is averaged within each pair of brain regions to yield the ROI level connectivity. Considering the high dimensionality of the connectivity features across different frequency bands and resting conditions, including eyes open and eyes closed, we employed a sparse clustering algorithm for simultaneous feature selection and clustering, as opposed to conventional clustering algorithms, which essentially treat all features on an equal footing. And these algorithms are therefore not able to correctly identify clusters when some of the features are redundant or noisy, such as the feature 2 here. By contrast, the sparse clustering algorithm is able to automatically zero out the redundant or the sparse clustering algorithm is able to automatically zero out the redundant feature or the noisy feature and automatically identify the essential features to perform the clustering analyses. So overall, this subtyping framework would allow us to derive neurobiologically meaningful subtypes in a fully data-driven manner. So applying our subtyping framework to the resting state EEG data of a PTSD study reveals 2 subtypes with distinct connectivity patterns. What I've shown here are the connectivity matrices of the 2 subtypes and the healthy control. And we can see that the connectivity patterns are highly similar between subtype 1 and healthy controls. but are very different between subtype 2 and the healthy controls. And this observation is confirmed by calculating the correlation coefficient between the connectivity patterns of the healthy controls and the two subtypes. The sparse clustering algorithm is also able to select features predominantly in the beta band eyes open condition as the main driver for the two subtypes. Performing t-tails between the connectivity patterns of the two subtypes suggests that subtype 1 has stronger connectivity in the frontal regions, but weaker connectivity in the parietal and occipital regions. The interhemispheric connectivity between homologous regions is also notable. Within this PTSD dataset, we also used multiple criteria to validate the derived subtypes. Both the CH criterion and the gap statistic indicate that setting the cluster number to 2 actually yield the most separable two subtypes. We also examined the stability of the subtyping analyses by assessing the consistency of the subtype assignment between the full sample and the subsamples. The stability is above 90% for each subtype and for both subtypes considered together, demonstrating the discovered subtypes are actually highly stable. To test whether the discovered subtypes can be replicated, we also applied our subtyping framework to the resting state EEG data of a second PTSD study. Unlike the first data set, which was collected in a cross-sectional study where there is no treatment involved, patients in the second study received the psychotherapy after their resting state EEG data were collected. And we separated this second data set into two cohorts. Cohort 1 was used during the initial submission of our paper, and then cohort 2 contained subjects who completed the study after the initial submission of our paper, and we used the cohort 2 for further independent replication of our finding. For cohort 2, again, our subtyping framework yielded two subtypes with connectivity patterns consistent with those in the first data set. And because patients in the second data set received the psychotherapy as the treatment, we're able to assess the clinical significance of the subtypes by examining the treatment outcome associated with each subtype. So linear mixed model analyses revealed that subtype 1 is actually more responsive to psychotherapy than subtype 2. In terms of both, and this is in terms of both CAPS-4 and CAPS-5, which are two versions of the clinician administered PTSD skills. And similarly for cohort 2, two subtypes were yielded with, again, with connectivity patterns consistent with all, with those all in the previous PTSD data sets. And the subtype status again predicts the responsiveness to psychotherapy, with subtype 1 responding much better than subtype 2. We then investigated whether the discovered subtypes can serve as potential transdiagnostic biomarkers for psychiatric disorders. To do so, we applied our subtyping framework to a depression resting state EEG data set, which was collected in a placebo-controlled randomized clinical trial for depression where a patient received either antidepressant or placebo. Quite strikingly, in this independent depression data set, again, we found two subtypes with connectivity patterns consistent with those in the PTSD data sets. And linear mixed model analyses revealed that the two subtypes actually differ in the response to antidepressant versus placebo. Specifically, while antidepressant is much better than placebo in subtype 1, the two arms fail to separate in subtype 2. Treatment response analyses also revealed that subtype 1 has significantly more responders than subtype 2 for antidepressant, but for placebo, there is no significant difference in terms of the number of responders between the two subtypes. Finally, we also used leaf dataset out validation to assess the generalizability of the subtypes across datasets. Specifically, we applied the cluster centroid based classifiers derived from subtyping analysis of 2 datasets to the third dataset. And then we compared the predicted subtype labels with those obtained from subtyping analysis of the third dataset on its own. The classification accuracy is above 89% across all data sets. This demonstrates that the subtypes are highly generalizable across data sets. Now, the first 2 biomarkers I just presented are derived cross-sectionally without referencing to any treatment outcome. In the third biomarker study, we discovered a treatment predictive EEG signature for depression. Conventional machine learning algorithms actually take a divide and conquer type of a strategy to do treatment prediction. Specifically, to mitigate volume conduction, they first optimize a set of spatial filters, essentially by assigning weights to different channels and then linearly combining them to yield the latent signals. And then the band power features of the latent signals are calculated, followed by a linear regression model that relates the band power features to the treatment outcome. The spatial filters are typically optimized via unsupervised loading approaches such as independent component analyses or principal component analyses. And the linear regression model can be estimated via ordinary least squares or sparse approaches like lasso. However, a major limitation of the conventional machine learning algorithms is that the spatial filters are optimized with cost function not directly associated with treatment prediction, and therefore these algorithms are not optimal in terms of treatment prediction. To address this limitation, we developed a new machine learning algorithm called SALSER for EEG-based treatment prediction. Unlike conventional machine learning algorithms where the spatial filters and the linear regression model are optimized separately with different cost function, in SALSER, the spatial filters and the linear regression model are now optimized under a unified convex optimization framework to minimize the prediction error. And SALSER is able to handle several important algorithmic challenges. The spatial filters are optimized in a supervised fashion to ensure optimality with respect to treatment prediction. And the number of latent signals is minimized via low rank regularization to avoid overfitting. And finally, the optimization problem is formulated as a convex optimization problem such that the global minimum solution is always guaranteed. So we apply SALSER to the pre-treatment EEG data from the EMBARK study, which is so far the largest neuroimaging coupled clinical trial for depression. And we found a signature predictive of the treatment outcome for sertraline, which is a widely used antidepressant. The correlation coefficient between the predicted and the true symptom change is 0.6. Applying the sertraline signature to the placebo arm does not predict the treatment outcome, suggesting this signature is specific to sertraline. The spatial patterns of the latent signals are predominantly in the parietal occipital regions and the frontal regions. We also compare SALSER with conventional machine learning algorithms in terms of the treatment prediction performance. These algorithms, as just described, use either band power features in the channel space or unsupervised approaches to extract band power features in the latent space. However, none of these approaches are able to find a signature predictive of the treatment outcome for sertraline. EMBARK is a multi-site study with different EEG systems across study sites. This actually allows us to assess the robustness of SALSER to different EEG systems by performing the leaf side out prediction analyses. Specifically, we train the machine learning models based on the EEG data from 3 study sites, and then apply the trained machine learning model to the resting state EEG data of the 4th study site. In this arguably worst case scenario where the EEG systems are different between the training and test set, Seltzer is still able to find a signature predictive of the treatment outcome for sertraline. Now, just to show how the sertraline signature can be used for treatment stratification, we did a median split on the predictive symptom change from the sertraline signature. Patients above the median are called biomarker positive patients, and patients below the median are called biomarker negative patients. And we found for biomarker positive patients in the sertraline arm, their response rate is much higher than the response rate without using any biomarkers. So this suggests that if we use the sertraline signature to decide who to get the sertraline as the treatment, the response rate can be substantially improved. We also validated the sertraline signature on multiple independent depression data sets. Data set 2 is from a naturalistic study where historical information about the treatment resistance to antidepressants is available. By applying sertraline signature to this independent depression data set, we found that the partial responders have greater predicted symptom change than the treatment resistant group. So this actually demonstrates the generalizability of our sertraline signature to the broader construct of treatment resistance to antidepressants. In our earlier study, we also established the task fMRI sertraline signature in EMBARK. So to test the convergent validity of the resting state EEG signature and the task fMRI signature, we applied them to another independent depression data set where patients were assessed cross-sectionally with both resting state EEG and task fMRI. On this independent depression data set, we found that the resting state EEG and task fMRI predictions are significantly correlated with each other. So this provide a convergent support for the existence of a treatment responsive neurobiological signature in depression across neuroimaging modalities. Finally, we also examined the relationship between the sertraline signature and the RTMS treatment in depression. Analyses were performed on a forced data set where patients received 1 Hz RTMS over the right DLPFC. By applying the sertraline signature to this independent depression data set, we found that last predictive symptom change from the sertraline signature actually is associated with greater response to RTMS. In other words, patients who are predicted to fail to respond to sertraline may actually be more amenable to the RTMS treatment. So this finding provides a potential evidence-based treatment selection approach for depression. So in conclusion, in the past, I guess 40 minutes or so, I have gone through our contributions in 2 aspects. We have developed tailored signal processing and machine learning algorithms to identify the spatial temporal patterns from EEG and to robustly decode EEG signals. And based on these algorithms, we discovered a number of replicable EEG biomarkers for subtyping and treatment prediction for psychiatric disorders on an individual level. Moving forward, on the basis of my prior research and by leveraging the Excellence Initiative at University of South Carolina, my plan is to set up a neuroinformatics and biomarker study lab to develop data analytics in collaboration with other faculty members here for multimodal data, including neuroimaging, behavior, cognition, as well as genetics. And then build and translate biomarker development platform for personalized treatment to psychiatric disorders. The lab will expand the scope of my previous research to different types of biomarkers and different types of brain disorders. My long-term plan is actually to establish a brain-based precision mental health platform. In this platform, we will use point-of-care biomarker tests to guide essentially every stage of the treatment process in the clinic, ranging from risk prediction, diagnosis, treatment selection, treatment monitoring, and follow-ups. So the success of this platform will critically depend on our ability to identify biomarkers at every stage. If established, this platform will allow us to get beyond the trial and error approach currently dominating mental health. In support of this long-term plan, in the short term, I will focus on methodological development for EEG-based biomarker discovery. In one, we'll be developing new methods for mapping brain circuits from EEG. I'll take a fundamentally different approach than what's been done in the field to estimate brain connectivity from EEG, which may substantially improve the performance of existing algorithms. In two, we'll be developing new methods for identifying EEG biomarkers for psychiatric disorders. I'll specifically address new data analytical challenges arising in the development of different types of EEG biomarkers, including the monitoring biomarker. And then finally, in three, we'll be developing methods for identifying the causal circuits associated with the discovered EEG biomarkers, especially for subtypes which may not be amenable to existing medications, but nonetheless may be better treated using brain stimulation therapies. And this may provide mechanistic insight into the EEG biomarkers and drive therapeutic development in the near future. Now, let me speak to the more details of these three aims. Conventional connectivity, EEG-based connectivity methods actually proceed in two steps. The first step is source reconstruction from EEG, which requires solving a highly ill-posed inverse problem. And then the second step is the estimation of the connectivity features in the source space, which may suffer heavily from spurious connectivity due to volume conduction. To address these fundamental issues of the current approaches, I'll develop new methods for mapping brain circuits from EEG based on our recent insight that connectivity estimation can actually be formulated as a multi-target regression problem. So this reformulation actually allows us to develop and assess algorithms under a principled machine learning framework. So within this multi-target regression framework, I will develop a spatial temporal variant of the autoencoder to learn the optimal mapping between the sensor space connectivity and the source space connectivity. I'll then propose data synthesis strategies to generate data to train the autoencoder. Specifically, I will leverage mathematical modeling techniques in conjunction with the prior knowledge we can gain from ECOG and fMRI connectivity studies to be able to simulate a large amount of data that are needed to train the autoencoder. And based on the connectivity estimation, I will develop new methods for dissecting the heterogeneity of psychiatric disorders, which may provide novel targets for the brain stimulation therapies. As I presented earlier, both disease subtypes and the treatment predicted biomarkers have been derived to parse the heterogeneity of psychiatric disorders. However, the disease subtypes were derived without referencing to any clinical information. And considering there are many ways of performing the subtyping analyses across different features and clustering methods, the pursuit of clinically meaningful subtypes is like a fishing expedition. By contrast, the effort to derive the treatment predicted biomarkers is too heavily dependent on the clinical symptoms, which may prevent us from fully capturing the underlying neurobiology. So to address these issues of the current approaches and to get the best sort of the both worlds, I will develop a semi-supervised subtyping framework for using clinical symptoms to guide the subtyping analyses. And within this framework, I will impose further constraints on the subtypes to regularize the solutions. The first is the modularity constraint, which assumes the connectivity patterns appearing in local clusters are more neurophysiologically plausible. The second is the label consistency constraint, which constrains the cluster labels to be identical across the repeated EEG measurements from the same subject. So this semi-supervised subtyping framework would allow us to derive clinically and biologically meaningful subtypes in a fully data-driven manner. Now, the EEG biomarkers alone do not directly inform therapeutics. To drive therapeutics, I will develop methods for identifying causal circuits associated with the EEG biomarkers. Specifically, I will use concurrent TMS EEGs to stimulate multiple nodes across the brain to be able to map the causal circuits. I will then relate the EEG biomarkers and the TMS EEG maps to be able to identify the causal circuit abnormalities. And then the identified causal deficit can then serve as a target for using repetitive TMS to remediate the circuit abnormalities. Based on the insight that these different modalities can be related via multi-view learning, I will develop a deep neural network to map the EEG biomarkers and the TMS EEG maps to a common latent space where the correlation can be maximized. I will then project the latent features back to the input space so to intuitively visualize the latent features. So this part of work will yield a principled approach for developing brain stimulation therapies based on the EEG biomarkers. Finally, I'd like to thank my advisors, collaborators, and co-authors for their tremendous help on my research and various grants for supporting my work. With that, I'd like to stop, and I'm happy to take any questions or comments.
Well, on behalf of all my colleagues here, greetings. Really enjoyed the presentation.
Thank you.
It was very effective. So let's just get started with some questions on the chat. Would you like me to read, or would you just like to read and answer as many as you can? You can start with the one with Ramtin Zand at the top.
So the question is, I'm wondering if the use of spiking neural networks and neuromorphic systems have been considered for recognizing EEG spatial temporal patterns, since they are known to be very efficient for these kinds of data sets. To my knowledge, I'm not sure if spiking neural networks or the neuromorphic systems have been considered. I do know there's a lot of deep learning algorithms that have been developed for EEG analyses in recent years. The main issue for EEG decoding, especially in the context of mental health, the issue is that the data is not. We don't have a lot of data to train these deep neural networks. So what have been found is the most effective is actually what is called the shallow network. Instead of using multi-layer neural network to do the decoding, we typically use 2 or 3 layers of network to do the decoding. And in that case, the salsa algorithm we developed is actually can actually be considered as a shallow network as well. And of course, there's other challenges like local minima with the deep learning approach that needs to be addressed in this case. And our work developed a convex optimization. We formulated the problem as a convex optimization problem, and I guess address that issue, the local minimum issue, in a sort of a new way. And of course, there are other ways of tackling these issues from other perspectives. So overall, I think there's a lot of interesting new perspectives we can bring in from the deep learning field in this respect.
I appreciate it. Thank you very much. I know there are many questions, so we can talk about it later in email or something. Thank you.
Yeah, sure. So the second question is, any qualitative label on subtype 2? I'm not sure what qualitative label means here. Are you able to clarify a little bit?
Yes. So what I was trying, so in subtype 2, some slides back, it was saying that there was basically antidepressant treatment didn't work. So what was there any label by, say, some medical expert on who are these types of people, or any insight at all besides just their raw neural imaging data on what are the characteristics of these data points as people?
Yeah.
That's what.
Yeah. So as I presented, there's actually, we look at the clinical difference in terms of the treatment outcome between the two subtypes, and subtype 2 actually is not amenable to medication, but this is something I didn't put in the slides. We also tested the two subtypes on another data set where patients received rTMS treatment. And we found that there is actually no significant difference in terms of the treatment outcome when both subtypes were getting the rTMS treatment. So in this case, subtype 2 may be better treated with brain stimulation therapies. And then in terms of the baseline symptoms, we also compared the two subtypes. There is actually no significant difference between the two subtypes in terms of the baseline symptoms, or even demographics are similar between the two subtypes. So so far the differences are mainly discovered within the neuroimaging data, not clinically.
Okay, thank you. That is very interesting. Thank you.
In the LEAP data set validation, do you group different data sets as discovery data? So in the LEAP data set out validation, we actually used we have three. I don't know if you refer to the subtyping analysis or the treatment predictive biomarker study. In the subtyping analyses, we have 3 data sets in total, right? And then every time we use 2 data sets to do the clustering, to do the subtyping analysis, and then tested the subtypes on the additional data set. So 2 data sets were used as the discovery data in this case.
Yeah, actually, let me clarify my question. So actually, yeah, it's the subtyping project I'm talking about. So I was actually wondering, how did you figure out the because the 2 discovery data sets actually some kind of batch effect. So did you fit the model with each data separately, or did you group or pull the data together? So how did you handle the batch effect with the 2 discovery data sets?
We actually put the 2 data sets together. When we trained the subtypes. Can you clarify what you mean by batch effect?
Yeah. So usually, because I'm a geneticist, so I'm very interested in your future goal to integrate the genetic biomarkers. So actually, in our research area, we will usually take the batch effect into consideration, because each data set may have different, because they are taking different time points, they are from different population. So it's more like we consider the group as a covariant.
I see. Yes. Yeah, I see. Yeah. So you're talking about the it's similar to kind of like the side effect we're talking about,
right?
In the treatment predictive study. Yeah. Interestingly, in the subtyping analysis, we didn't do anything to deal with the side effect, and we still are able to get good results. So which is a little surprising to us. I mean, it's definitely worth dealing with the side effect in this analysis as well, to see if we can get even more improvement in terms of the leaf data set validation performance. In the treatment predictive biomarker, when we do the leaf data set out prediction, or the leaf site out prediction, we actually use the site variable as a covariant beforehand to regress out the side effect, and then do the prediction analyses. So that's how we dealt with the side effect in the treatment predictive biomarker study. But yeah, in the subtyping analysis, we didn't specifically deal with the side effect.
Okay, thank you. Nice talk, by the way. I look forward to meeting with you.
Thank you.
Well, there's some more questions. We can go for one or two, but otherwise, let's see. There's there's one long question from.
So got it. So the question is about the TMS stimulation. What is the amplitude? We used 120% of the motor threshold, and for how long and at what frequency? So we used so for identifying the causal circuit response, we actually used the single pulse TMS. So it's not repetitive TMS. Single pulse is actually the interval between different pulses is actually 2 seconds. And then how many pulses did we apply? We applied 60 pulses per stimulation sites. And then for the RTMS treatment, because I presented several RTMS findings in our work, the frequency we used are either 10 hertz or 1 hertz. For the PTSD, it's actually 1 hertz. And then for depression is both 10 hertz and 1 hertz. 10 hertz is over the left DLPFC, and then 1 hertz is on the over the right DLPFC.
All right. There's one more question, but we don't have to do that right away. It says, can you elaborate on causal circuits? And let me formally, though, thank you. There are a lot of comments here saying fantastic talk.
Thank you.
And anybody wants to drop out can drop out. I'll just spend 1 or 2 more minutes with Vi before we officially close that. Thank you very much, everyone.
Thank you.
Mike, you are welcome to stay. So if I understand correctly, by the way, how did your voice held up?
I mean, it's still okay, but you can definitely tell, right? Yeah, it's unfortunate.
Tomorrow, if I remember correctly, at 3 o'clock, you have a meeting with the dean.
Okay.
He's a great guy, Hossein Haj Hariri. If possible, I want to carve out some time. It can be any time, amount of time, half an hour, maybe a little more. Mainly not for you to talk, but mainly for us to exchange information that will be helpful in your decision making. For me to understand any special issues to think about, in terms of your how good a fit this particular position is. Share with you some a little bit unusual aspects, or different aspects than just the basic faculty position that we may have, so that a typical department may have. So
Okay.
that's generally the idea, and having that background would be probably helpful. So can we find some time? One possibility is to look for the time available between your availability tomorrow at noon and your availability, at 3 o'clock. The other option is I may be available, or I'm available today later on also. I think I have a meeting until 3 o'clock. Let me check if I can. Yeah, I'm free between 3 and 4:30, or yeah, kind of thing. So just in case this is Eastern time. So just in case that is available for you, we can meet that. But otherwise, I'll talk to Randy, who's scheduling the meetings, and we'll see whether there is any time between noon and 3 before you have meeting. And it's not absolutely necessary. It's just nice to have kind of thing.
Yeah. I have, I think 3 p.m. Eastern time should work for me today.
Okay.
Yeah,
So
I have, yeah,
So,
okay,
kind of open-ended. We can talk for a few minutes, or a little longer,
okay.
whatever, and mostly focused on your questions, or helping you just better understand this particular position, this
Yeah,
particular.
I appreciate that. Yeah.
Okay, I'll send
Sounds
you a
good.
link that you can call. Mike, if you're still listening, I'll send you a copy in case you have time. You're most welcome to join. But
Okay, I will.
all right, great. So let's then we'll talk at 3.
All right, thank you so much.
Thank you.
All right, talk to you later. Bye.
Bye.